Hello and welcome to DoveBiology Bio Lessons to Go. In this video, we'll be exploring the chromosomal theory of inheritance, which is covered in AP Biology, Chapter 15. With advancement in microscopy, scientists were able to determine that the inheritable factors that Mendel was describing in his experiments with pea plants were actually genes which were carried on chromosomes. Today, we know that humans have over 20,000 genes, which are found on 46 chromosomes. These genes have specific locations that they're found on these particular chromosomes. These locations we call loci. We can actually tag using fluorescent dyes genes to be able to observe their locations on those particular chromosomes. For example, in this image here, we've tagged a pair of genes on homologous chromosomes with a yellow fluorescent dye. Now, because of the Human Genome Project, we've been able to determine the location of many of our genes throughout our genome. We've also discovered that each chromosome may contain thousands of genes. When genes are inherited on the same chromosome, they are said to be linked. Certain characteristics seem to be inherited together more frequently than others, which indicates that perhaps they are linked together on the same chromosome. For example, the inheritance of red hair with freckles. Now, normally, when genes are on separate chromosomes, they can sort independently of each other during meiosis. This is Mendel's principle of independent assortment. In this particular example, we have 4 chromosomes, each with a specific allele being identified, heterozygous for the B trait and heterozygous for the R trait. Because these alleles are on separate chromosomes, they can sort independently of each other. The dominant B allele can sort independently of the R alleles, so we can get the capital B with the capital R, or we could have our capital B with a lowercase r. The recessive B allele similarly can sort independently and go with our dominant R trait, or it could go with the recessive R trait, allowing for 4 different combinations in our gametes. It's kind of like if 2 individuals are traveling in separate cars. Because they're in separate cars, they can go in separate directions. They can sort independently of each other. But when alleles are on the same chromosome, they tend to stay together during meiosis. It's very difficult for them to separate. So because the dominant alleles in this case are on the same chromosome, they will tend to go together in this case. Or the recessive alleles will tend to go together. It's kind of like being in the same car going on the trip. You can't quite easily go in separate directions. You have to go where the car goes. Now, the idea of genes being linked together was discovered by a scientist by the name of Thomas Hunt Morgan. Now, he discovered that certain factors were inherited along with gender, which indicated that these alleles were probably linked to the sex chromosomes. Now, he discovered this while working with fruit flies, the Drosophila melanogaster. Now, fruit flies, like peas, have easily distinguishable traits. And while working with fruit flies, he discovered that there were a number of mutant varieties that had specific inheritable patterns. One pattern that seemed to interest him was the inheritance of this white-eyed characteristic. Now, normally fruit flies have a red eye trait, and this white-eye characteristic seemed to be inherited in specific ways that were linked to gender inheritance. So he assumed that this factor was probably carried on the X chromosome, and we can actually observe this in a Punnett square. So a male fruit fly would have the X chromosome and the Y chromosome, and the allele for eye color would then be carried on the X chromosome. So we indicate that by doing a superscript on our X chromosome. So the X and the Y aren't actually alleles, but actually whole chromosomes. And the little superscript represents the allele that is connected to that particular chromosome. The female receives two X chromosomes. And if it is a carrier for red eye trait, we would have a red eye gene, which allows them to be red eyes, and then it would carry the white eye trait. So when we allow them to cross, you have four different possibilities. You could have a pure red-eyed female who only has red-eye genes. We can have a red-eyed male, a red-eyed female, but then you could have the white-eyed male. Because the males only receive a single copy of that allele, there's nothing to mask the white-eye trait. So the white-eye trait is what shows up. And so because of this particular pattern, this indicates that we have a trait that is linked together on the sex chromosome. Since their discovery in fruit flies, we found that there are a number of sex-linked traits that are also found in humans. These include things like hemophilia, red-green color blindness, male pattern baldness, and Duchenne's muscular dystrophy. Sex-linked recessive traits have particular patterns of inheritance. For example, if the father expresses the trait and the mother is normal for the trait, then all of the children will be phenotypically normal. They will not express the trait because the mother's dominant allele can mask the recessive allele that was passed on by the father. If the father is normal phenotypically, but the mother is a carrier, there's a 1 in 4 chance that one of their sons will receive that allele. Since the mother passes the X to the offspring and the father gives the Y, and there's nothing on the Y to mask that allele on the X, there's that 1 in 4 chance that the offspring would have it. Now, if both the father expresses it and the mother is a carrier, then this is the only way that a daughter can express that X-linked and recessive trait. In order for a daughter to have that X-linked recessive, they have to get the recessive allele from their mother and their father. And this is only possible if the father has the trait and the mother carries the trait. So if genes are found on the same chromosome, is there ever an opportunity for them to separate? Well, the answer to this lies with meiosis and the process of crossing over, which was discovered by Alfred Sturtevant while working with Thomas Hunt Morgan in the fruit fly room. To observe the process of crossing over, Sturtevant crossed a heterozygous female for 3 linked traits with a recessive male for the same traits. Now, if these linked genes never separated, the Punnett square predicts that 50% of the resultant offspring should have the dominant trait and 50% should express the recessive. But after completing the cross, he actually did not observe this. He saw that about 10,000 fruit flies, about 2/3, either showed the dominant or recessive, but the remaining actually showed a mixture of the dominant and recessive traits in different amounts. Sturtevant concluded that the only way that these alleles could have separated is through crossing over in the X chromosome during meiosis. So that as a result of crossing over, we can get a new combination whereby the recessive body and eye color trait could be inherited along with the dominant trait for wings. And the dominant body and eye color could be inherited with the recessive trait for minute. Crossing over results in additional genetic combinations when genes are linked, and this allows for greater diversity in the offspring. It appears that crossing over is more likely between genes that are further apart on the chromosome because there's greater opportunities for those homologous segments to separate from each other. For example, the characteristic of eye color And wing size is more likely to separate than, say, body color and eye color because of their greater distance on the chromosome from each other. Now, the distances between these genes can be expressed as map units. One map unit, or centimorgan, represents a 1% recombination frequency. Genes that are further apart on the same chromosome can have a recombination frequency near 50%. Such genes are physically linked, but genetically unlinked and behave as if they're found on different chromosomes. In other words, they cross over quite frequently. To observe or compare the chromosomal makeup of an organism, a karyotype can be prepared. Metaphase chromosomes are going to be stained and photographed. Those photographs are enlarged and the individual chromosomes can be cut out. We then match up the chromosomes with their homologs and line up the chromosomes from biggest to smallest. The last pair of chromosomes are going to be our sex chromosomes, and all of the other chromosomes we call autosomes. We can compare those chromosomes based on their size, their shape, their centromere location, and the banding pattern when we stain with specific stains like chiasma stain. Examination of a karyotype allows for us to identify the presence of possible chromosome mutations. Chromosome mutations are changes in whole chromosomes which are going to impact multiple genes and traits. This is different from a simple gene mutation, which could produce a change in one gene that affects a singular trait. Examples of chromosome mutations might be a structural modification of a chromosome as a result of like a genetic breakage, or it could be the presence of an irregular number of chromosomes as a result of non-disjunction. During crossover, chromosomes may break or reassemble incorrectly, causing changes in the genes that are present on those chromosomes. These changes in chromosome structure could cause a genetic disorder. One example of a chromosomal mutation would be a deletion, whereby a whole chromosomal segment would be removed. Like in this particular case, segment D has been removed. And so along with that would be any and all genes that are associated with that particular segment. Perhaps a homologous segment will reattach back to its original source, leading to a duplication. When that chromosome gets inherited, it would have additional genetic information. An inversion might occur where that segment, when it reattaches, attaches in reverse. Or perhaps a translocation will take place where that segment that is broken off will actually reattach to a non-homologous chromosome somewhere in the genome. An irregular number of chromosomes will occur as a result of non-disjunction. Non-disjunction occurs when the chromosomes don't separate equally during anaphase of meiosis. If the chromosomes don't separate equally during anaphase 1 of meiosis, none of the resultant gametes will have an appropriate number of chromosomes. All of them will either have too many or too few chromosomes. If non-disjunction happens as a result of anaphase 2, it's possible that at least half of the resultant gametes will have the appropriate number of chromosomes, whereas those that are the result of non-disjunction will have an abnormal number of chromosomes. Inheriting too many or too few chromosomes can lead to many disorders. An incorrect number of chromosomes is referred to as being aneuploidy. If you only inherit one copy of a given chromosome, that's referred to as being monosomy. And in humans, there's only one survivable monosomy condition, and that's monosomy X, where you inherit only one X chromosome, because that's all one might need for survival. Whereas with their autosomes, we have to inherit two copies, one from each parent. If you inherit 3 copies of a given chromosome of any pair, this will be referred to as a trisomy disorder. A common trisomy disorder is trisomy 21, referred to as Down syndrome. Now, some organisms are said to be polyploidy. They have more than 2 complete sets of chromosomes. This is pretty common in plants, but rare in animals. One of the only animals that is considered to be polyploidy is like a Chilean squirrel. Being polyploidy, you can be triploidy or have 4 sets of chromosomes. Strawberries are actually octoploidy. They have 8 sets of chromosomes, which is why we select those as an excellent organism for DNA extraction, because they have so many copies of DNA inside of their nucleus. Upon examination of a karyotype, we can determine if an aneuploidy condition exists. For example, this young lady has an extra chromosome on number 21. She is experiencing Down syndrome. Now, these conditions could actually be diagnosed prior to the birth of an individual by removing cells from the amniotic fluid or cells of the chorionic villus and preparing a karyotype. By determining whether or not an abnormal chromosome number is present, it allows for parents to make healthcare decisions for their unborn child. The human genome consists of 3 billion bases, which are organized in our 46 chromosomes. Of those 3 billion bases, only about 1.5% are responsible for our 20,000 genes. The remaining sequences are non-coding RNA sequences, DNA transcription factors, introns, and sequences that we've yet to find out what they do. More study of the human genome is necessary for us to have a better understanding of our genetics.